All right, welcome to the March meeting of the Ann Arbor chapter of the American Statistical Association. We're very lucky to have a speaker today, Debraj Bose, who is a PhD student in the Department of Biostatistics at the University of Michigan. His advisors are Mike Benke and William Wen. His dissertation focuses on two main areas of statistical genetics: evaluating the performance of aggregation tests using analytic calculations and extensive simulations based on real data. And two, developing a method to prioritize causal genes for human traits and diseases by integrating both genome-wide association study, or GWAS data, molecular phenotypic data, and gene features as well. And he also collaborates with the metabolic syndrome in men, the METSIM study. And thank you for letting in, Walter. So thank you very, very much for giving us a first peek at your wonderful soon-to-be job talk.
Thank you, Susan, for the very nice introduction. And thank you, everyone, for attending online and also in person.
Could you maybe use a microphone so that it gets
that's what I was
to be audible on the for people on Zoom?
Can people hear me better?
Is it on?
Yeah.
How about now?
Yeah.
Someone wrote something in the chat too. I don't know.
Yeah, someone wrote something in the chat.
The sound was good. Oh,
Thank you, Walter. And thank you, everyone, for attending my talk today, both in person and on Zoom. I'll be presenting on two different projects from my dissertation on rare variant association testing, as well as causal gene prioritization in human genetic studies. But before we get into the meat of all of that, let's start at the most fundamental level, which is cells. The human body is made up of trillions of cells, and almost every one of the cells contains a nucleus. If we took a deeper look, we'd find that our DNA is packed into 23 pairs of chromosomes, where one out of each pair is inherited from each parent. And then if we looked even deeper, we'd find that the human genome is composed of 3 billion base pairs, where each position you have one out of 4 nucleotides: adenine, thymine, guanine, and cytosine. And at positions throughout the genome, we might have individuals carrying different bases. These are what we call genetic variants. And to understand how genetic variation influences complex human diseases and traits, we often perform a GWAS, or a genome-wide association study, where we test every single genetic variant throughout the human genome for association with your phenotype of interest. And usually you'd plot results from every single variant test that you're performing in a GWAS in the form of a Manhattan plot, such as the one on this slide, where the strength of association, typically measured as negative log 10 p-values, for each single variant test performed throughout the human genome is plotted. So GWAS have given us the ability to scan through the entire genome and look for disease associations. But there are still some challenges that remain, and my dissertation focuses on two of those challenges. The first one is that GWAS are typically underpowered to detect associations with rare variants. And we define rare variants as those with minor allele frequency, or MAF, of less than 1%, where minor allele frequency is defined as the percentage of chromosomes in the population carrying the less common version of the variant or the less common allele. So to address this, researchers developed what we call aggregation tests, where instead of testing each individual rare variant separately, you'd combine the rare variants within a gene and test the combined effect of all these rare variants on your trait of interest. And despite the promise of aggregation tests, in practice, we've seen over the years that single variant tests have still found more associations than aggregation tests, even for rare variants. So we thought it was imperative to perform an investigation into finding scenarios, basically sample sizes and aggregation tests, sample sizes and genetic models for which aggregation tests are expected to be more powerful than single variant tests, because we now have much larger growing biobank sample sizes. And we published this paper, sorry, go ahead.
That's all right. Just a simple, very simple question.
Go ahead.
I understand rare is less than 1%. Is there a kind of a median of what's a common variant? Is that on the order of 2 or 5 or 10 or 20% for common variants?
That's a good question. I probably should know the answer to this. I actually don't.
Maybe it's an ill-posed question. Maybe it depends on I don't know.
It could be anywhere between 1 and 50.
I see. Okay.
Yeah. And usually if a variant is common enough, then you'd be easily able to detect associations in a GWAS. I'm sorry that I cannot answer your exact question.
That's good enough. I understand now. Okay, thank you. That's good.
So in past studies where we'd have even lower sample sizes, people would define rare as less than 5%. But now that we have large enough sample sizes, I think the general consensus is to define it as 1% for rare versus common.
Yeah.
So so that's what I'll talk about in the first half or first part of this talk. And we also published this last year in American Journal of Human Genetics. And in the second part of this talk, I'll talk about Project 2, which focuses on a post-GWAS challenge. So GWAS identifies significant genomic regions, but actually turning those GWAS hits into biological insight remains a challenge because it's hard to pinpoint the true causal gene. And that could be due to a variety of reasons. Most often it is due to what we call linkage disequilibrium, or LD, which refers to the correlation between variants. So variants that are closer to each other in the genome are usually inherited together, and therefore they are highly correlated. So if we zoom into this particular region in the Manhattan plot, you might be able to see something like this, where due to LD, a lot of the times the most significant variant might not even be the true causal variant, and it could be any variant or variants in this particular genomic region. And also notice that there are about 9 or 10 genes in the region, so it's really hard to actually pinpoint the true causal gene or causal genes. More than 90% of GWAS hits are in the non-coding region of the genome. And so these regions regulate gene expression in quite complex and context-specific ways. So it's really hard to get to a single gene from hits in the non-coding region. So to address this, researchers have tried to integrate other lines of genomic evidence. And one such important example is that of a molecular QTL study, or quantitative trait locus, which is defined as a genetic variant that is associated with a molecular phenotype or a gene product like gene expression and protein levels. And I'll try to explain this a little bit better. So if you remember from Brady's talk last month, he spoke about the central dogma of biology, which basically states that our DNA undergoes a process called transcription and gets converted to messenger RNA. And the levels of messenger RNA produced by a gene is called the gene expression levels. And then messenger RNA gets converted to polypeptides or a chain of amino acids, which eventually become proteins through a process called translation. So these are what we would call the protein levels produced by the gene. So if we have a genetic variant that influences gene expression or protein levels, and we examine this variant and we find that it's also associated with our phenotype of interest, that might give us a sense of the underlying biology or the underlying mechanisms through which these GWAS signals or GWAS hits act and then eventually get to our phenotype. So project 2, or the second half of the talk, we'll be talking about developing a method called MOSAIC to integrate genetic association data, molecular QTLs, as well as gene features to prioritize causal genes from GWAS significant loci. I'll go into project 1 unless there are any questions, and I'll also stop between project 1 and 2 for any clarifying questions that anyone might have. So going into project 1, the project is titled Rare Variant Association Studies, when are aggregation tests more powerful than single variant tests? This project was published last year in the American Journal of Human Genetics. If you're interested to look at the manuscript, here's a QR code. And this project was advised by Mike and Christian. So as I mentioned before, single variant GWAS historically has been known to have low power for detection of associations with rare variants. which is why aggregation tests like burden, scat, and scato tests have been developed to increase power to detect associations with rare variants. And in this project, we asked the question for what range of genetic models and sample sizes are aggregation tests expected to be more powerful than single variant tests. And we answered this question for a normally distributed trait following a linear additive model. So just a brief background on aggregation tests before I talk about how we actually answered this question. Aggregation tests pool association evidence across multiple rare variants in a gene instead of looking at them individually. And then some common aggregation tests are burden, scat, and scato, and we'll be investigating these three and comparing them with single variant tests in GWAS in this project. Aggregation tests require the selection of a mask to specify which rare variants to include, with the aim to include causal variants in the mask and include the likely non-causal ones. And so to that end, we choose the most likely functional variants, like protein truncating variants and missense variants. So if you think of the central dogma and if you think of the gene as an instruction manual for making proteins, and proteins are what perform the most important functions in our bodies, a protein truncating variant is a variant that will cut or alter the protein that is being produced by the gene, so it's going to result in a loss of function. So it's quite a very harmful variant. And missense variants will result in a change of one amino acid in that sequence of amino acids that's going to be produced by the gene. So in some cases, these could be very benign. In some cases, these could be extremely disruptive or deleterious. And in this project, we also focus on, we only focus on rare, defined as minor frequency less than 1% variants. And so I'll talk about how we create a gene mask for aggregation tests. With this toy example of 25 variants in our gene of interest. So a single variant test in GWAS would basically perform 25 separate tests for association of each individual variant with the phenotype of interest. But if we were to do an aggregation test, we would first use a software like Variant Effect Predictor to functionally annotate the variants and pick out the missense variants and the protein truncating variants. We would then look at the minor allele frequencies and pick out the rare variants out of these ones. And in this example, we find that these 6 variants satisfy both our functional annotation criteria as well as the minor allele frequency threshold. And so we would aggregate these 6 variants in our PTV plus missense rare mask. And different aggregation tests will have different ways of aggregating these 6 variants within the mask and then testing for association between the phenotype of interest and the gene as a whole, represented by aggregation of these 6 variants. So we first performed analytic calculations to compute power of single variant, burden, and scat tests. We did not attempt analytic calculations for scat-O because no closed form solution exists for power, and we'd require numerical integration. So under some simplifying assumptions, we were able to show that the non-centrality parameters of the chi-square statistics and hence the power for all the tests depended on the product of sample size and heritability, the number of causal variants, and the total number of variants in the gene. And when I say heritability, I basically mean the proportion of variation in the trait that can be explained by genetics alone. And so we fixed some of these parameters. So we fixed product of sample size and heritability at 50 and the total number of variants at 50. And then we varied the number of causal variants from 1 to 50. And we plot power curves for single variant burden and scat tests. And we find that at a smaller proportion of causal variants, single variant test had greater power, whereas for a larger proportion of causal variants, burden test had greater power. And normal cases, we'd also observe an increase in power for scat with increasing number of causal variants. But due to the simplifying assumptions that we made, we actually happened to stumble upon a very special and interesting case that we found where change in proportion of causal variants did not impact the power of scat. And so those were just one particular choice of the parameters, which is why we made an R-Shiny app for our analytic calculations, which you can access at this QR code and link, where we let the user investigate parameters like sample size, heritability, and number of variants. And we've listed in the simple case tab all the simplifying assumptions we made on top of the variants being independent and looking at power curves of burden, scat, and single variant test. We also made a general case tab where we relaxed those simplifying assumptions and only had the assumption of the variants being independent, where on top of sample size and heritability, you could look at different effect size models, aggregation test weights, as well as use our inbuilt minor allele frequencies or upload your own set of minor allele frequencies for variants in a gene that you're looking at to obtain power curves. So we, after performing our analytic calculations, we next proceeded to do simulations with UK Biobank exome data, where we had 6.7 million rare biallelic variants for 378,000 unrelated white British participants. For computational purposes, we only restricted ourselves to chromosome 2, and we investigated 3 nested masks, the smallest of which contains only protein truncating variants within a gene. The intermediate mask contains protein truncating variants as well as missense variants that are annotated to be deleterious by 5 different algorithms. And the biggest mask contains PTVs, deleterious missense variants, as well as all other missense variants. And we analyze the 1,060 genes in chromosome 2 that have all the 3 masks present in them, because these 3 masks are nested. And for the sake of time in this presentation, I will show you one result with the PDV plus missense deleterious mask. But if you're interested, you can look at our paper for other results as well. And in our simulations, we focused on a range of values for sample size, heritability. And instead of looking at proportions of causal variance, now that we have 3 different functional annotation categories, we look at probability of being causal for PTVs, deleterious missense variants, and other missense variants. And the only result for this part of the talk that I'll show is the effect of changing sample size from 30,000 to 100,000 to 300,000 at a heritability of 0.1% for a gene, and probabilities of being causal for the 3 different categories at 80%, 30%, and 1%. And when we perform aggregation tests with the intermediate PDV plus missense deleterious mask, these box plots represent the distribution over the 1,060 genes in chromosome 2 that I showed in a couple of slides ago, of the difference in power between the corresponding aggregation test and single variant test. So the blue box plots would be basically power of burden test minus power of single variant test over 1,060 genes. Similarly, yellow represents the same for scato, and then red the same for scat. And the numbers on top of each box plot denote the proportion of genes for which the corresponding aggregation test is more powerful than single variant tests. So the higher up a box plot is over the line at 0, the more genes for which aggregation test is more powerful. And similarly, the lower a box plot is below the 0 line, the more genes for which single variant test is more powerful. And we find that at smaller sample sizes of at a smaller sample size of 30,000, aggregation tests are more powerful than single variant tests for over 80 or even over 90% for one particular aggregation test than single variant tests. And then when we kept increasing sample size, of course, power for every test increases with increase in sample size, but we find that at a sample size of 300,000, single variant tests are more powerful for a large majority of the genes, and aggregation tests are more powerful for barely 3 to 9% of the genes. So the main takeaway from that plot would be that with rapid growth of large-scale biobanks, we would expect power for single variant tests to increase more rapidly than aggregation tests. And not shown in these slides, but another conclusion we found with the PDV plus missense deleterious mask was that with a sample size of 100,000 and heritability of 0.1%, aggregation tests with that particular mask are more powerful than single variant tests for a majority of the genes, when at least 80% of the PTVs, at least 30% of the deleterious missense variants, and less than 1% of the other missense variants are causal. And if you're interested, you can find results for the other. We find situations like genomic thresholds for these causal probabilities for the other masks as well, for which aggregation tests are more powerful for a majority of genes in the paper, as well as looking at different parameters like effect size models, aggregation test weights, as well as direction of effect. So the conclusion would be that even though we do find more situations where single variant tests are more powerful than aggregation tests, we would still recommend using both single variant tests as well as aggregation tests together, because we did find, and we highlighted them in our paper, different genetic models and sample sizes under which aggregation tests also were more powerful than single variant tests. So that is project 1, and I'll pause here if there's any clarifying questions. Yes.
I was wondering if, when I was looking at the boxes in the box plots, it looks like the width or the height, however you want to say that, the height of the box plots are bigger in the center with the 100,000. Is that just am I interpreting that as a variability or not? Should there be scaling for variation or not as sample size changes?
So this was just plotting the distribution of the difference in power
Oh,
over 1,000 simulation replicates for every gene. So each of these box plots correspond to power estimates, in power estimates for 1,060 genes. And I think this is just what we observed in our simulations.
okay, yeah.
Yeah, but that's an interesting
I was just
observation.
expecting, I didn't know if it's a measure of variability or not, so.
There's a power curve you usually see with studies that, you have very low power and then there's sharp changes in power. And then their power is high and kind of levels off again.
Right.
So the variability in power, that kind of matches that idea,
Okay, yeah, so
right?
variability
So the
is
middle
what
group is where the power is shifting the most.
Right.
Yeah, okay,
Yeah.
I see.
Yeah.
So at lower sample sizes, aggregation tests are a lot more powerful than single variant tests. And so you see, for most genes, that is true. So you see that for most genes, it's within that small like width of the box plot. But then in the middle is when aggregation tests, or rather single variant tests, start to shoot up as well. And then at the end, both tests are quite powerful, but single variant tests are probably just a little bit more powerful.
I see. Thank you. That helps interpret that. Thank you.
It's much better than what I said, since you were doing differences.
Yeah. No, but that's what you said made me exactly think of that. Yeah.
I have a question.
Yeah.
I should know this, or maybe you said it, but so the heritability is fixed at 0.1%.
Right.
How did you draw the effect sizes, the variance?
The effect sizes
People have-
right. So the question in person was that the heritability is fixed at 0.1% for a gene, but then how do we come to our effect sizes of the variance from this heritability? So the answer to that is we chose effect sizes proportional to negative log 10 of minor frequency for causal variance. So that basically means giving larger effects to more rare variants and then giving smaller effects to less rare variants.
And so did you, could they be either positive or negative?
So we, in these particular simulations, we chose them to all be in the same direction, so all positive.
Right.
But in the paper, we've also looked at the other direction.
Because obviously burden assumes that.
Exactly, yeah. So as to what Brady said, burden test is expected to be more powerful when all the variants affect the trait in the same direction. And that's what I'm showing in this plot right here. So I'm giving burden test its best possible scenario and then basically still saying that we still find more associations with single variant test when we have this large sample size. And then in the paper, we look at the other extreme as well when we have variants completely in the opposite direction, like 50%, 50%. So yeah, if there's no more questions, I'll proceed to project 2, where we develop a method called MOSAIC to integrate genetic association data, molecular QTLs, and gene features to improve causal gene prioritization. This project is advised by William and Mike, and it's still work in progress. So I'll really appreciate any feedback that you might have. So as I mentioned in the introductory slides, we identify significant genomic regions from GWAS, but not really the causal genes, due to various reasons such as LD or the correlation between variants masking the true causal variant, as well as the fact that most GWAS hits are in the non-coding regions, so associated loci may not contain protein coding variants, so it's hard to map them to coding genes. So the question is, how can we integrate multiple lines of genomic evidence on top of genetic association results to prioritize causal genes? And so researchers have attempted to design various gene prioritization strategies over the past decade and a half. One type of strategy integrates GWAS association data with molecular QTLs. And just as a reminder, QTL, or a quantitative trait locus, is a genetic variant that is associated with a molecular phenotype or a gene product like gene expression or protein levels. And so methods like TWAS, colocalization, or intact attempt to either find correlations between the phenotype and imputed expression or imputed protein levels, or establish if overlapping GWAS signals and molecular QTL signals, if they're in the same genomic region, whether they're due to the same shared causal variants. Another more recent strategy published a couple of years ago integrates GWAS association data with this giant list of gene level features that they've created. to compute a polygenic priority score for every gene. So this POPs method leverages polygenic enrichments of over 57,000 gene-level features from diverse sources such as disease pathways, protein-protein interaction networks, or even from publicly available expression datasets. And so the goal of our project was to develop a method which we eventually named MOSAIC, which stands for Multi-Source Statistical Aggregation for Identifying Causal Genes, to integrate GWAS association data with molecular QTLs, as well as gene-level features. And so the way we do it is we first allow the user to use any integrative method of their choice, like TWAS, colocalization, or INTECT, to integrate GWAS association data with molecular QTLs to obtain gene-level output in the form of z-scores or posterior probabilities, which we then convert to base factors for every gene. We then integrate these base factors with these gene-level features from the POPs method to obtain our MOSAIC posterior probabilities of being causal for every gene, which we can then use to get like a ranking of genes and look at the top however many genes that we want to. And I'll slowly walk you through how we statistically formulate the problem. And the different colors in this slide will denote the different components. So first we have the X's, which are our gene features. And in blue, I denote the prior. So these gene features, we assume encode some prior information on whether a gene is causal or not. And the gammas denote latent binary causal status indicator variables for the genes for the given phenotype. And then we assume that we see evidence of this causality through gene-level output from any method that integrates genetic association data and molecular QTLs. So the red is going to denote our observed likelihood component. And this likelihood component could be of the form of z-scores or posterior probabilities, which we will then convert to base factors. But what we eventually want to find is this posterior probability in green. That is the probability that a gene is causal or gamma equals 1, given our gene features or the prior information, and given D, or the gene-level output from any genetic association data and molecular QTL integrative method, which is the likelihood component for all genes. And so if we break down this probability a bit more, we can write it from the graphical model as proportional to a prior component, probability of gamma equals 1, or probability of a gene being causal given our features, times a likelihood component, or probability of gene-level output from any integrative method given that a gene is causal. And so the most natural assumption to make for our prior is a logistic model, where we need to estimate our logistic model parameters alpha and betas. And we can also write our likelihood as a base factor by dividing by probability of D given gamma equals 0. So our required posterior can be written as probability of gamma equals 1 given x comma D as is proportional to our prior component probability of gamma given x, which we can obtain from our logistic model after estimating alpha and betas, times the base factors which we can obtain from the observed likelihood component of our model. And so two things to keep in mind are that we still need to estimate alpha and beta in the presence of latent variables gamma. So we'll use NEM algorithm. And also we have more than 57,000 features for roughly 20,000 genes. So we have more features than the number of genes. And that's something that we really need to keep in mind when we fit a model. So how our method works is as input, we will use the gene feature matrix that the POPs authors created. So this is going to be roughly a 20,000 versus 57,000 matrix for gene versus features, as well as the base factors that we obtain from any method that integrates genetic association data and molecular QTL data. And since we have so many features, we would, in practice, expect only a small proportion of them to be actually relevant for the phenotype that we're interested in. So we do a marginal screening step where we reduce our set of features by fitting marginal models for each feature, converting regression p-values from these marginal models to q-values, and then retaining features with a q-value less than 0.05. And if you're not familiar with q-values, q-values are used to control false discovery rate. And the interpretation of this last line, retaining features with q-value less than 0.05, would basically mean that in the retained set of features, we would expect on average about 95% of these to be actually relevant features, and then 5% of them to be noisy or false. And then with the reduced set of features, we go into our EM algorithm step where we did some math, and then ultimately we found that our E step boiled down to updating our posterior probabilities, and the M step simplified to updating our prior parameters alpha and betas by fitting a logistic model with ridge penalty with the current posterior probabilities as the response and the matrix of retained features as the predictors. And the ridge penalty is essential for cases where we might have more features than the number of genes, so we need some sort of shrinkage. So we iterate the E and M steps till convergence to obtain estimates of alpha and beta hat from our prior model. And then we put those, plug those estimates into our formula for the posterior to obtain our MOSAIC posterior probabilities of being causal for every gene for the given phenotype of interest. So before I show you results with MOSAIC, I want to quickly compare our method with POPs and show you how the two methods differ. MOSAIC explicitly allows for molecular QTLs to be integrated, whereas POPs does not. MOSAIC, both methods assume that the gene features encode prior information on whether a gene is causal or not. We assume a logistic model for that, whereas POPs assumed a linear relationship. We estimated the prior parameters iteratively with an EM algorithm. POPs did it in one step by fitting one single linear model. Both methods also used a marginal screening step. We used a Q-value threshold of 0.05, whereas POPs used a p-value threshold of 0.05. As method output, we provide posterior causal probabilities for every gene, whereas POPs provided what they call priority scores for every gene. And they also discuss in their paper that these priority scores are not often going to be really comparable or transferable across traits, whereas you could compare probabilities across traits. So we simulate 5,000 normal features for 20,000 genes. We set true values of alpha and beta in our logistic model for the prior, where we set beta to be very sparse. And by that, I mean we only allow for 100 features to have non-zero betas. The remaining 4,900 features will have zero betas, or rather beta set to zero. And then for the betas that we allow to be non-zero, we simulate them from a normal distribution centered at 0 with variance sigma square, where we vary sigma over a range of values to look at different strengths of priors. At lower values of sigma, since we simulate betas to be centered at 0, these simulated betas will not be very meaningfully different from 0, so the prior is going to be extremely weak. Whereas once we get to larger values of sigma, we'll have more and more betas that are meaningfully different from 0. So these are going to be more informative and stronger priors. And so that's one component for our model, the prior component. For the likelihood component, we need base factors. And so we get to those by first simulating causal status for the binary causal status for every gene. And then based on the causal status for a gene, we are going to simulate z-scores, which are going to emulate the gene level z-scores that you could get from like a TWAS or another method that would integrate genetic association data with molecular QTL data. And then we're going to convert them to base factors. So to simulate our z-scores for non-causal genes, we simulate them from a standard normal distribution. And for causal genes, we simulate them from this other normal distribution. And rest assured that we did not just make up these numbers. We wanted them to emulate real data, and we estimated this distribution from magma z-score distribution. Magma you can think of as just another aggregation test like a burden scat or scat-o test. So we estimated this distribution from a magma z-score distribution for publicly available schizophrenia summary statistics in the POPs paper's GitHub repository. So I'll show you results for our method, but first I'm going to show you how marginal screening works with Q-value versus P-values. And I'm going to argue in this slide how screening based on Q-values leads to a better signal-to-noise ratio than P-values. So I have the simulation parameters again in the bottom left for your reference. And this table shows you the average number of features selected over 100 simulation replicates by MOSAIC, which uses a Q-value-based marginal screening approach, and versus POPs, which uses a P-value-based marginal screening approach. And these two rows denote the average total number of features selected versus the average number of true non-zero features selected in each simulation over the 100 simulation replicates. Remember that we had 100 non-zero features, so you want to see how many of those actually also passed, not just the total number of features. And the standard deviation refers to the standard deviation of the normal distribution from which the 100 non-zero betas from the prior logistic model were being simulated. So the 2 points I want to make from this slide are that MOSAIC's Q-value-based filtering selects roughly 95% true non-zero features on average out of the total features selected. So if you look at the top 2 rows here, if you divide the top row by the bottom row, you'll see we roughly get about 95% true non-zero features selected on average. Whereas POPs, since it uses a p-value less than 0.05 as its criteria for including features, will select by random chance 5% of the 5,000 features, so about 250 on average every simulation replicate. And then it does select more non-zero features, but then it also selects way more extremely noisy features to go into the model. So screening based on Q-value leads to better signal-to-noise ratio for our method. And then POPs, we argue, has a worse signal-to-noise ratio. And this is going to be one of the contributing factors in the next slide, where I show that our method has better performance than POPs, other than the fact that we estimate the parameters iteratively over in EM algorithm, whereas POPs does it in one step. And also another thing I do want to point out is that at lower values of sigma, our method selects almost no features to go into the model, or very few features, because at lower values of sigma, these estimated feature effects betas will be extremely low. They'll probably not be very distinguishable from zero. So our method recognizes that it, or correctly identifies the situation that we have a very weak prior, and so it doesn't really put in any of that weak information into the model, whereas POPs by default will put in 5% of the features. So in simulation results, we compared MOSAIC with POPs and Bayes factors. We looked at the mean optimal F1 score. F1 score is the harmonic mean of precision and recall, and it's pretty commonly used in the machine learning world to investigate the classifiers. And we see that we also have these points in blue, which are the oracle prior, and these points in yellow, which are the oracle posteriors. So the oracle prior and the oracle posteriors are 2 theoretical quantities that we thought would be interesting to see in simulation, since we know the true values of alpha and beta. So we could plug in these true values in the formula for the prior and the posterior for every gene, and just see what the strength of the prior is compared to the likelihood, as well as what the posterior would be had we been given the true values of alpha and beta instead of having to estimate them in using MOSAIC. So first conclusions are that when sigma is low, that is when the effect sizes are too small to distinguish from zero, the Q-value screening from the previous slide I showed typically selects no features. So posterior and base, so MOSAIC posteriors and base factor metrics are pretty aligned, whereas POPs gets flooded with noisy features and therefore exhibits poorer performance. But once we have sigma getting larger and larger, the effect sizes also get larger. And notice that the blue points keep getting higher, which means that the prior contribution is getting stronger, so the prior is getting stronger. And hence, MOSAIC and POPs, since it incorporates this prior information in their models, performs better than the base factors, which do not incorporate the prior information in the model. And the last thing to note is that even though MOSAIC does not reach the heights of the oracle posterior, its performance is not too far off from the oracle posterior, which represents a sort of a theoretical upper bound or a gold standard for performance of our method. And we also looked at mean area under the curve of the ROC curves, but results were pretty similar. So I'm not going to spend any more time on this slide, and rather I'll talk about the real data analysis that we conducted in the METSIM study, which stands for metabolic syndrome in men, which is a study of middle-aged and older men from Kuopio, Finland. We focused on 558 lipid metabolites out of almost 1,400 metabolites that were measured on 10,000 whole genome sequenced individuals, just because we usually observe many more associations with lipids than other kind of metabolites. And remember that our method has a likelihood component and a prior component. So for our likelihood component base factors, we are going to integrate genetic association data from GWAS on the 558 lipids with protein QTLs. So we are going to integrate 558 lipid GWAS individually with UK Biobank estimated protein levels from pQTLs for about 2,600 genes using INTACT, which is a method that was developed by one of William's students. And also, we are going to convert these intact posterior probabilities for the 558 lipids for each gene. So each gene now has 558 different intact probabilities. So we are going to collapse those into one individual probability for every gene, which can be interpreted as the probability that a gene is causal for at least one lipid. And for the prior component, we are going to use the POP's full list of features, 57,534 features for the same 2,600 genes. And we are going to use base factors that we'll compute from the intact probabilities on the left and the feature matrix on the right to obtain the MOSAIC posterior probabilities. And here I have the intact posterior probabilities versus MOSAIC posterior probability scatter plot. Just keeping in mind that intact integrates GWAS association data on the lipids with pQTL data. And then our method integrated intact probabilities, so both these genomic sources along with gene features. So we found that overall, both methods seem to be very much in agreement, except for these two interesting genes over here. The first one is the ACOX1 gene, which encodes the peroxisomal acyl CoA oxidase enzyme. It's also involved in fatty acid metabolism and lipid homeostasis. And the other gene, the LPL gene, which is involved in fat breakdown, energy and storage, and metabolic health. So two pretty well-known lipid-related genes that intact wasn't really prioritizing, but then MOSAIC prioritized by explicitly modeling gene features. And if you were interested in what kind of features MOSAIC really picked out and ranked towards the top, out of the top 50 features with largest estimated effects from the EM algorithm in MOSAIC, we found more than 30 of those features fell into one of these 6 buckets. So they were either involved in fatty acid and phospholipid metabolism, peroxisome biology, or lipid-related pathways, liver and gut, mouse and human expression principal components, PPI network genes that reinforced lipid biology, and pathways of phenotypes common in mouse knockouts of lipid metabolism genes and peroxisomal disorders. So the good thing, we were really happy to see that even though we literally threw in a tsunami of features at MOSAIC, it was really able to pick out the relevant ones, in this case, relevant for lipids, and really rank them towards the top and make them drive the posterior probabilities or drive the prior component of our model, and then really find out these two genes that were lipid related. So the takeaways from the second part of this talk is that we propose MOSAIC, which is a flexible statistical framework to integrate multiple lines of genomic evidence to prioritize causal genes. In simulations, we showed that MOSAIC outperforms the POPs method, which was the first method that aimed to integrate association data with their giant list of gene features. And in real data application in the medicine study, we showed that explicitly modeling these gene level features enabled prioritization of plausible causal genes by MOSAIC beyond those identified by existing integrative approaches that did not explicitly model these gene level features. So in conclusion, I showed you two different strategies today for linking GWAS signals to causal genes. First, aggregation tests that were developed because GWAS were underpowered to detect associations with rare variants, could also be used to elevate genes as candidates by demonstrating that rare genetic variants within the gene collectively impact the phenotype. And you could integrate GWAS results or aggregation test results with other lines of genomic evidence like eQTL, pQTL data, or gene level features, and gene level features using our method MOSAIC to improve prioritization of causal genes, and MOSAIC provides a very general statistical framework to do that. With that, I'd like to thank my advisors, Mike and William, as well as Christian and other members of my dissertation committee for their guidance and support, as well as Laura and other members of Benki and Scott group, and other mentors and friends who've helped me along the way. And thank you all for listening, and I'd be happy to take questions.
That was a absolutely fantastic talk.
Thank you.
I would hire you for this.
Thank you.
Yeah, not only is the science really cool, but you made pretty nice toys for people to implement it. They look fun. Some of you probably know all the answers, but would you like to toss a question out?
I'm beginning to think we're the only three members of the audience who can act. That's good questions. For some reason,
Ask some questions if you want.
I have a question, but
Go for it.
Oh, please go for it.
okay. In your test, when you were comparing a mosaic and pops, and you were looking at the chart, and you were saying, and I was thinking about the table earlier when you said that, you had like it was more precise on or filtered better, and of the ones that it picked, they were the correct ones.
Right.
But I think you said that there were 100 to find.
There were 100. Yeah.
So what my question is, if Pops found an additional 20, would you be able to test those 20 in Mosaic, or is it not set up to test those additional ones that Pops might have found? Could you use Mosaic to test what something is found, or is it just, once you get the results, you can't go back and
That's a good question. So in our simulations, we actually know which ones are the true non-zero features and which ones aren't. So we were able to actually see that Pops was able to get 20 more, but in real life, we won't really know which ones are the true, correct features that should have been identified. So
there would be no way of telling.
There would be no way of telling. So that's, unfortunately, yeah, we couldn't really use Pops for that, or the Pops marginal screening step for that purpose for our method.
Thank you. All right, that makes sense.
It's a similar question, but could you just use the features that you identify with Mosaic in Pops, or is there?
I think we did. I don't have it on these slides, but Pops performance definitely increased because the signal to noise ratio got better. Mosaic was still better because of this, because of the fact that we do the iterative estimation of the prior parameters versus Pops doing the just in one step. And also we felt that actually this was William's suggestion to keep Pops' implementation like what the author suggested instead of really changing it there.
You don't want to make theirs better.
Yeah.
So how does you integrate the molecular QTLs through just using intact?
Right. Yeah.
So what does Pops what is the input besides the features?
You mean in the field analysis or?
In general, because you can't just obviously input the same
For Pops.
For Pops, yeah.
Yeah, you wouldn't do that. So you'd do it completely separately. You would take your GWAS summary statistics, you'd run Magma gene-based test, you'd get the Magma z-scores, and then you'd run Pops with the Magma z-scores, get a completely different set of.
So you can only use z-scores.
You could only use z-scores. And
Magma z-scores?
That's what they say in their paper. And their z-scores are kind of, I think we've spoken about this in group meetings, but their z-scores are kind of one-sided. So the z-scores that are shifted towards the right are the more significant ones, whereas z-scores that were left were actually the non-significant ones. They do it in a very weird way. So you couldn't really use two-sided TWAS z-scores in this case.
I think I have to toss you a question.
Yeah, of course.
That anybody could ask and therefore has a high chance, a high prior probability, let's say, of being asked at a job talk. And that is, you've got all of these sensitivities and specificities from your ROC curve. Have you thought about positive and negative predictive value for people who have these rare variants, predicting that they're going to end up with these problems?
That's
Government might be interested in such a question. I'm just.
I mean, in gene prioritization method, that's always a big issue. Like, how do you validate your method, or how do you really say that, okay, these are like gold standard genes? Because truly, like, such a gold standard set of genes doesn't really exist. Everyone just makes their own kind of list or their own kind of evaluation criteria, which the POPs paper also did. So,
So I think that the default kind of answer is if you're testing for a condition that's pretty prevalent in the population, your positive predictive value is going to be much better, right? And so when you have rare variants and rare phenotypes that you're trying to associate them to, you're kind of in a bit of a fuddle for using it to predict which people prospectively will be at risk for these various conditions, but you do get a better look at the science for what's causing these things.
exactly.
And because you use this word screening in your talks all the time, you're going to have a natural tendency for a brain to go through screening criteria using. So you see what I mean? It's very easy to hop to these types of questions since you've already put the word screening in their head. You got ROC curve stuff on your slides. And so my prior probability is that this would be a very easy question to ask.
And also, people use polygenic risk scores more for that kind of an application, where they would, aggregate associated variants across the entire genome, and these would be like very significantly associated and independent variants, and then get like a score by aggregating all of these variants to see how likely it is that a person with a particular genetics would get a certain disease or have a certain disease in the future. So that's another slightly related but also different application area.
Okay, I don't have any other easy questions that someone like me could come up with.
Let me ask you another easy question about your first project.
Oh, sure, go ahead.
I know the answer. When you defined deleterious missense variants, did you think about using alpha missense to
So,
variants?
good question. This is before alpha missense, I think. Yeah, we got this published before all of that was a big thing.
And then someone might also ask if you thought about including mass with splice variants.
So the definition of PTVs, I don't have it on the slides, but it includes stop gain, start lost, spliced variants, frame shifts.
All splice?
I think
Some
maybe we can go back and check.
like splice AI, where they
Oh, no, probably not. No, definitely not that. No. Yeah.
like sometimes in like non-canonical splicing regions.
No, not those. Whatever VEP, variant effect predictor, provides.
I don't think it existed if anyone asks.
Yeah.
You were doing this. So.
All right, let's thank our speaker again for an excellent talk. Thank you.